To study the dynamics of individual molecules over an extended period of time, molecules must first be localized in space. We accomplish this by immobilizing them on the surface of a microfluidic cell. The procedure starts with the assembly of the cell, which contains 4 chambers where the biomolecules will be fixed. The chambers are connected to the exterior through inlet and outlet capillaries. The cell is made by fusing a patterned PDMS disc to a quartz cover slip. The assembled cell is then mounted on the fluorescence microscope, and flexible tubings are connected to the capillaries to allow the easy exchange of solutions through the chambers. To activate the surface for molecule immobilization, first flow a solution of biotinylated BSA through one of the channels. BSA will adsorb to the chamber surfaces. Then, flow a solution of streptavidin, which will in turn bind to the biotin. Finally, flow your biotinylated oligonucleotide, which will bind to the streptavidin, thus anchoring the molecule of interest to the surface of the quartz cover slip. An imaging buffer, designed to maximize the lifetime, stability, and intensity of the fluorophores, is flowed through the chamber at a constant rate throughout the entire acquisition period. The automated setup scans different sections of the surface for molecules having both donor and acceptor fluorophores, and then moves them one by one into the probing volume, obtaining for each a fluorescence intensity time trace until both fluorophores photobleach.
Hi, I'm Nicolas Fiori from the laboratory of Professor Meller in the Biomedical Engineering Department at Boston University. Today we will show you a procedure to obtain single molecule FRET traces from immobilized nucleic acids using